This is where we left off. I want to say a little bit more about the way DNA is constructed, or RNA. So far, we know that DNA is a huge polymer, the biggest polymer like that life makes, millions of individual monomers big, that the individual monomers are known as nucleotides, that a nucleotide in general is made out of a phosphate and a sugar and a base, or a nitrogenous base, that the sugar for DNA is called deoxyribose, which is where DNA gets the D at the beginning of its name. We know that even though DNA is made out of millions of nucleotides, those nucleotides are only 4 different types because there are 4 different nitrogenous bases that are found in DNA. And those 4 bases are Gattaca, right? G, A, T, and C. We know that DNA is double-stranded, and it's double-stranded so that the one strand and the other strand fit together the way a piece of pottery fits into its mold. I want to say one more thing about the structure of DNA, which is that the individual monomers get put together so that the sugar and the phosphate, that'll be the phosphate, that'll be the sugar, are connected to the next sugar phosphate, are connected to the next sugar phosphate, are connected to the next sugar phosphate, sugar phosphate, sugar phosphate, sugar phosphate, making up what often gets referred to as the backbone of a strand of DNA. Or you could refer to it as like the handrails on like the spiral staircase of the DNA, right? And then branching off towards the middle are the bases. See, G, A, T, and C, Gattaca. The bases are branching off towards the center there. So that a base pair, the step, is made out of the nitrogenous bases, whereas the phosphate and the sugar are joined together by covalent bonds. Oh, here's another important thing. Oh gosh, going to be a question undoubtedly. The phosphate and the sugars are put together with covalent bonds. Covalent bonds are the strongest bonds, but here in the middle, do you see how they've got those little dotted lines like here between the C and the G? Those dotted lines represent hydrogen bonds. So when it comes to the structure of a molecule of DNA has got lots of covalent bonds that are connecting the phosphates and the sugars of these nucleotides to each other to make up this backbone or handrail. Branching off towards the center are going to be the nitrogenous bases, and the nitrogenous bases are joined to each other in the middle with hydrogen bonds, the weakest of the bonds, but there are millions of them there in the middle, so DNA gets held together pretty well, kind of like Velcro, right? All right. Now, there are rules of complementarity. If anyone asks you, what are the binding rules? What are the rules of complementarity? What are Chargaff's laws? I won't ask you that, okay? What they mean is when it comes to this piece of pottery and this mold fitting together, how do they fit together? And there really are things that are kind of like ridges that go together with things that are kind of like grooves. And there's 2 different kinds of ridges and 2 different kinds of grooves, okay? I call the ones ridges. These are the larger molecules. This would be a purine, and the smaller ones are pyrimidines. All right, so the rules of complementarity. An A always binds to a T, and a G always binds to a C. Here's how I remember it. C and G, they're both the round letters, okay? All the time, whether it's DNA with DNA or DNA with RNA, the C always binds to the G. With DNA, the 2 angular letters, the A and the T, they bind to each other, right? Now that's going to be very, very useful. Let me just erase this. Okay, so you can think of a groove here. This will be like an a thymine, okay? And over here will be an adenine, right? The ridge fits into the groove, okay? So adenines fit with thymines, cytosines fit with guanines. So these two would bind together and these two would bind together. Great. Okay, so now we got the setup. DNA has got two big jobs to do. Remember that DNA is like an entire library filled with books, and each of those books has got chapters in it, and each chapter is going to be the instructions to make a protein that will make one of you. Okay? The reason that you are different from someone else is because you've got different proteins and they got put together in the way that makes you. All right? So DNA in general is enormous. It's like an entire library. Now, job one. Job one is known as replication. Job one is known as replication. And replication is every time one of your cells divides, it goes from being a single cell to now being two cells. Well, when it started off as a single cell that only needed one copy of DNA, if it's going to be two cells, well, it's going to need another copy. So we don't trust anyone just to write things down longhand. We have got a system for making a perfect copy of all of your DNA. And that perfect copy uses the binding rules of complementarity. Here's what's going to happen. What's going to happen is this molecule called topoisomerase is going to help the strand of DNA that is a spiral staircase. It's going to unwind it and hold it in the open position for a moment. Then there is a molecule named helicase, and helicase is going to break the hydrogen bonds that are holding together the adenine with the thymine and the cytosine with the guanine. So here comes helicase, and it's going to break them, okay? So you started off with this, then it unwound by topoisomerase, and then helicase is going to split it apart. Now, when it's split apart, now I've got adenine, cytosine, guanine, thymine, blah, blah, blah. And over here, I've got the complementary ones, and they're away from each other, all right? Now, there is going to be an enzyme. Remember enzymes? There's going to be an enzyme, and this enzyme's name is DNA polymerase. DNA polymerase is a great name for this enzyme. Why? Because A's means it's an enzyme, and the rest of the name is supposed to tell you what it does. And DNA polymerase, right here, DNA polymerase makes a polymer of DNA. So what does DNA polymerase need to do? The only thing DNA polymerase needs to do is it needs to look at, let's do blue. It needs to look at that and decide which base that is. And if that base is an A, it will put a T. Okay, so here we've got our DNA polymerase, and the DNA polymerase is like, oh, that's an A, here's a T. That's a C, there's a G. That's a T, there's an A, that's an A, there's a T, that's a G, there's a C. That's all he has to do. Easy job, right? That's all he has to do. How is he going to make a mistake? First of all, it's really easy. I mean, I've told you the rules of complementarity. You're going to be using them in lab. And if I say, wherever there's a C, you put a G. If there's a G, you put a C. And with the same thing with AT, how are you going to make a mistake? But that's not enough, all right? You can't put two A's together because two A's together would be like trying to bump two ridges together. They're too big, they won't fit. So it always has to be a ridge with a groove, right? So it always has to, it would either have to be that or it'd have to be that. So we'll make it even easier though. The C and the G, they fit together. They've got three little hydrogen bonds that fit together, all right? The A and the T, they only have two. So this little guy, he's an enzyme. Enzymes are not the smartest robots in the world. This job that he has is if he sees an A, he puts a T. And if he makes a mistake and sees an A and puts another A, they don't fit together. Sees an A and tries to put a G, they don't fit together. Sees an A and tries to put a C, they don't fit together, okay? It's really, really hard for him to make a mistake, all right? Now, on top of that, even if he does make a mistake, there is another enzyme that's going to follow that enzyme and double check his work. And if he's walking down the strand and he says, "Oh, Bob, what were you thinking? You put a G next to a T," and he'll fix it. So DNA's got two big jobs. This job is going to be called replication. Replication only happens once in the life of a cell. The cell will say, "Okay, I've gotten so big, I think I'm going to divide now." It will make a perfect copy of all of its DNA, put it into two different nucleuses, and then boom, divide the cell so that now each cell has got a perfect copy of DNA. Here's something that I would like to point out to you, okay? I want you to notice that this stuff, that's the existing DNA. This stuff, that's the DNA that will go to daughter cell number 1, and this will go to daughter cell number 2. The Okazaki fragments and stuff, I don't hold AP120 responsible for, okay? I'd like you to notice that each daughter cell gets one of mom's original strands of DNA and one new strand. Do you see how one of mom's original strands of DNA went here and one of her original strands of DNA went there? And then the rest of the DNA is new strand. Since each of the resulting strands of DNA with DNA replication, since each will be made out of one original strand and one that's newly built, that is why we call this semi-conservative replication. Semi-conservative, because each strand is made out of one original strand and one new strand. All right. So now you can see that this is really a brilliant answer to how do you have information that needs to be copied trillions of times in your lifetime and never make a mistake when we couldn't even copy this page 30 times without getting mistakes inside of the document, right? Your cells can do this trillions of times and never make a mistake because life with a capital L has made it so easy to do it right and not make mistakes. Now, the problem is in order to make this so error-free, so easy to make right, we have whittled it down to just 4 bases. The 4 bases, from the point of view of that enzyme there, the 4 bases are like black, white, red, yellow. Like if I told you black and white go together, red and yellow go together, ready, set, go, you're not going to make a mistake because they're so different, right? But here's the problem. The problem is DNA, this is the library, and inside of it are chapters in books that have instructions for how to build a protein. Well, proteins are not built out of just 4 amino acids, are they? Proteins are built out of more than 20 amino acids. So now we have got this system that works great, okay? But there are only 4 unique things, and I need that to be able to tell the ribosome how to build a protein that's made out of more than 20 different amino acids, okay? We're going to get to that in a moment. So that's DNA. The job number one for DNA is replication, but that is something that DNA only participates in when the cell is going to divide like this, DNA replication, all right? When the cell is going to divide, and now it is going to have two different cells, and each one will have a perfect copy of all of the DNA to make a perfect you, all right? We will start here at the beginning of the next lecture.